I've I started out in the lab doing computational work, and then started to do experiments this year. So in totality, my project this year has been working to develop a model to assess fluid composition using acoustic levitation by yours truly. So we've said in my work in the lab, we've worked on ultrasound, using ultrasound to work with biological fluids. And in general, ultrasound is used a lot in clinical practice and in biological research. Probably one of the more popular examples of the use of ultrasound is imaging, and because it's not invasive and it's not harmful to biological tissue. Probably the most popular example of that is imaging of the fetus during pregnancy. But they've also started to use ultrasound for even more invasive techniques like destruction of cancer cells. Ultrasound, they're trying to use ultrasound to precisely target cancer cells and not harming other cells, which has been one of the hard things about cancer treatment in general. So those are some common uses for ultrasound in biology. But our lab in general, we use ultrasound to study blood coagulation. And the reasons why we're still alive, even though we've had cuts and wounds before, is because our blood coagulated when that happened. The cells clump together like this here, and with proteins and platelets and other components, they slow down the blood flow so you don't bleed to death. And there are methods of measuring blood coagulation like this one. It's the rotational thromboelastometer. But one of the main problems with such devices is that they require sample contact with the device. And that could lead to inaccurate measurements and unwanted chemical reactions that might alter measurements. So our lab has developed technology to where we can use ultrasound to perform non-contact measurements to mitigate that kind of problem. And here's an example of levitated fluid. How it works is we have a transducer and a reflector, and that transducer sends acoustic waves, and the reflector, it bounces off the reflector, and we create this acoustic field here. And at nodes in that acoustic field, the acoustic force balances out with the force of gravity, and because of that, you can levitate objects there. So that's how we levitate fluid here. We have two function generators. The first function generator sends the signal to the transducer to perform to make this field so that you can levitate. But then we have another function generator that sends another signal, and that modulates the field. And we do this over a range of frequencies. So that kind of applied force, we want to see how levitated droplet responds to different types of forces, and we vary that force by frequency. So, and we see how the drop responds, and that drop oscillates because every kind of component, everything in nature has a natural frequency. And so if we have a frequency, if we send a frequency that is equal to that natural frequency, the drop is going to resonate. It's going to oscillate more than at other frequencies. And we measure this by having a light source and a photodiode, and then we put the drop in the path of that light. And so as the drop oscillates, it blocks some of that light. So the more it oscillates, the more light it's blocking. And we record how much the drop oscillates via that method. So we talked about, we have resonant frequencies, we have natural frequencies. When we have a frequency where we send a force, and if we have the frequency of the force matches the natural frequency, then we have resonance. And different material properties contribute, they determine what a resonant frequency of a particular sample is. And so the hypothesis is we want to leverage those differences in resonance in order to identify differences in fluid structure and composition for diagnostic purposes. So that's the hypothesis of this whole project. So to test that, we have this example picture of a levitated sample. We decided to make a model, using a control, which is a 5% dextrin solution. And then to kind of represent cells, although they're not cells, they don't move like cells, but they are small like cells. We had some polystyrene microparticles of 8 micrometers in diameter, and we put them inside of the control, the dextrin solution. And then we observed the spectra of those different solutions. And of course, here we have different levels of microparticle concentration. We have the control, we don't have any microparticles, and then we put more and more microparticles of different molarities of microspheres in the solution. And then we levitated them and we observed spectra. So this is an example of a signal that you'll get from a drop oscillation. And what we do is this is a signal in the time domain. We apply a fast Fourier transform to move from the time domain to the frequency domain. And we can use this to observe which peaks are most resonant. So we do this for the different types of samples we've levitated. And in order to observe if we have any differences, we look at the spectra of different ones and we compare. And for the most part, this is what you get. So here, this graph kind of highlights the fact that with the more concentration, we have a 0.1 molar of polystyrene microparticle solution. That one, the one where we had the highest amount of microparticle concentration, that gave us a second resonance peak compared to the other experimental groups that were levitated. So that made that special. So from this, you might think, well, it's over. We figured out the problem. More microparticles, more resonant peaks. We were able to detect that difference. But science is not that simple. So one thing that we observe from experiments is that over time, resonance shifts. The resonance changes with time. So as you can see here, we have different curves of, this is one of the control samples that we levitated. And we saw that here we have the darkest color here. This is when it began, but this is about 9 minutes later. This gray curve here, the resonance is far different from what it was, and the amplitude has been dampened. The amplitude is a lot lower. So we wonder why that's the case. And this has consequences because even in the microparticle solution where we had resonant peaks, we had 2 peaks. The peaks kind of, the resonance shifted and then they got absorbed. It's like the 2 peaks became 1. So the answer is not as easy as saying, well, more microparticles means more resonant peaks and can determine that structure. There are other factors that we have to understand in order to see what's going on. So in order to investigate why this is happening, we go back to kind of the basics, go back to the scientific basics that you learn in your general science courses. What causes resonance shifts? And resonance frequencies are determined by material properties. Material properties like viscosity and elasticity are pretty important, especially when we're dealing with fluids. So this is an example of viscosity and what that looks like. As you can see, this orange fluid flows at a lot slower rate than this blue fluid here. So the orange fluid is more viscous, and you could see that materials like honey are more viscous than water. So that's just an example. And then you have elasticity, which is the ability for an object to return to its position after it's been deformed, like a spring. And some fluids are viscous and some fluids are viscous and they're elastic. So this adds another level of complexity in what we're studying. So we want to look at, to observe this, what kind of changes are happening during the experiments. Well, for one part, one thing that we can observe from taking photographs of the levitated objects is that the droplet becomes smaller with time. So we can look at, obviously the drop is evaporating if that's the case. So from there, we can look at evaporation and humidity. This is the kind of factor I'll talk about today. There are many factors that contribute to this, but this is the one that's studied in this thesis. And this is a big topic right now because we have a pandemic going on and people are hopeful that this airborne virus is going to dissipate, at least for temporarily in the summertime, because it's more humid. When the humidity is higher, evaporation is less, and so the first droplets will carry through the air, not as much as if the humidity was lower. So that's just an example of evaporation and humidity. And so with the data, thankfully, I recorded the humidity on each day that the experiments were done. And then from there, we plotted the differences in humidity, what happened with the resonance shift. So on this axis here, we have how many, in terms of frequency, how far was resonance shift, how many frequencies did it shift per minute? And then here we have the different groups, and we can see with all of the groups, when there was lower amounts of humidity, and the humidity was lower, the resonance shifted more. This is the case for the primary resonance peak. So this occurs in the biggest peak, the first peak, because all of the samples had at least one resonance peak. And so that primary resonance is what's covered here. But then we have a second peak. And so two of the groups exhibited two peaks in the sample. So this is the second peak that's being observed. Although the resonance was only observed in, I think, two of the groups in this group. And then in this group, we had all of them, we observed two resonance peaks. But even in this case, with the lower humidity, you have more of a resonance shift. And so even with that in mind, it's important to note that these experiments were not designed to test for humidity. This is just an observation. So I wouldn't even really call this a robust scientific study, but from this kind of preliminary information, we can make a hypothesis, another hypothesis. And we could say that lower humidity leads to more evaporation. And because that lower humidity leads to more evaporation, the evaporation changes the concentration of different particles in the droplet, which leads to a shift in resonance. So in future work, we'll talk about how we plan to study that moving forward. So this is just a model. Dextrin and polystyrene microparticles are not plasma, and they're not red blood cells, and they're not other cells that you find in blood. So it's important to kind of talk about how this translates to blood. And blood is one of those fluids that is both viscous and elastic. And that's mainly because of red blood cells. So we have different kind of things in blood. You have your red blood cells, you have your white blood cells, you have your plasma, you have your platelets, you have all these things. You might have some other proteins and hormones that travel through blood. But red blood cells are the primary reason why you have elastic properties, because red blood cells have abilities to deform so they can fit to tight spaces so that you can get the oxygen to your tissues. This is why diseases like sickle cell anemia are so terrible, because it limits those red blood cells to deform and squeeze in those spaces. And of course, we talked about the red blood cells' ability to clump together when we were talking about coagulation. And so that contributes to the viscous effect and the elastic effect that blood has. That's why they call it a viscoelastic material. So if we can understand through experiments how viscosity and how elasticity, how that changes resonance and how that impacts resonance shift, then we can use that to better study, red blood cells in terms of the amount you have in your blood, whether you have abnormalities. So understanding viscosity and elasticity are extremely important. So to conclude what we have here in the study, it's one thing we want to kind of point out is, again, it's not as easy to say, we have more microparticles, therefore we have another resonance peak, that's it. There are so many things that are impacting these experiments, even though the idea of the analysis and the thesis wasn't to understand or directly test any of these effects. Those effects are important. And so that means we have to make new experiments that are designed to directly measure the effects of humidity and the effects of viscosity and elasticity and other fluid properties on resonance structure. But even so, we can kind of see that evaporation of droplet during experiments is a problem. And so one of the things that we'll be looking at, especially because I'm going to return for a master's here, so that's one of the things that we'll be looking at, is designing shorter experiments while still gathering the same information so that we can mitigate those effects of evaporation. And so once those things are taken care of, we can also look at kind of how the spectra itself. We can look at if there are information that we're not getting from these experiments. And one thing that's the deal when you're interacting with ultrasound is that there is some backscattering. So when a sound wave interacts with a material, that material is going to, the properties of that material are going to cause some of that sound to reflect back. And they use that in imaging when they're doing ultrasound imaging. The different kind of tissues in your body reflect that ultrasound wave differently. And so they use that information to make images. And so kind of maybe this is the same thing is happening in this levitation experiment. Maybe you have different types of cells and different types of things that happen in blood that they're reflecting, but we're not measuring. They're reflecting, some of that sound wave, but we're not measuring that. So that's another thing that I think that I would like to try to do moving forward with these. So that concludes my honors thesis defense presentation. Of course, I have to acknowledge Dr. K and how he's mentored me and helped me throughout this whole process, starting in the summer of 2018. I'd like to thank Dr. Brown and Dr. Naguib for being on my committee. I'd like to thank the students in my lab, most specifically Nithya Kasareddy. She was the graduate student who brought me into the lab and taught me how to levitate, improve my coding skills, really taught me the science of how all of this works. So this stuff is not possible without her and her help. It's not possible without, Scott Hummel, and even, how he's helped me with some of the coding for these projects as well. And of course, all of the undergrads who I've been in the lab with for the last 2 years, thank you. I'd like to thank the Tulane Department of Biomedical Engineering. I'd like to thank the Newcomb Tulane College, who gave me the grant in order to perform these experiments. I'd like to thank my parents too, because they encouraged me to stay in biomedical engineering and encouraged me to pursue an honors thesis. So thank you.